as food is broken down into smaller pieces, the salivary glands release saliva into the mouth. Saliva is approximately 99.5% water and 5% electrolytes and proteins. The most important proteins within saliva are amylase, which begins the digestion of carbohydrates. The second important protein is mucus. Mucus aids swallowing by moistening the food particles and holding them together. It also provides a lubrication to help the transport of the food bolus move from the mouth down to the stomach. Finally, an antibacterial action of a lysosome is to destroy certain bacteria by taking down their cell walls. It also rinses away any material that may be a food source for bacteria. Saliva is also rich in bicarbonate buffers. This neutralizes food as well as acids produced by bacteria in the mouth. Without these bicarbonate buffers, dental cavities would be more present. Saliva is triggered to be released when chemoreceptors and pressure receptors within the oral cavity respond to the presence of food. They can also be stimulated without oral stimulation, just by thinking about food, seeing, smelling, or hearing the preparation of food. Both the parasympathetic and sympathetic innervation of the salivary glands result in an increase in saliva production. Parasympathetic stimulation produces a watery saliva rich in enzymes. Sympathetic stimulation produces a smaller amount of saliva that is thick and rich in mucosa. This would be the cotton mouth that we would experience when working out or during stressful situations. The esophagus also produces and secretes some mucus. This mucus serves as a chemical and mechanical protector for the cells within the esophagus and to help propel the food bolus down into the stomach. The only digestion that occurs within the mouth is the start of digesting carbohydrates. Amylase, released in the saliva, starts to break down polysaccharides into maltose, which is a glucose-glucose disaccharide. Other than this, no digestion begins in the oral cavity. Absorption does not occur within the oral cavity for any nutrients. Since the stomach serves as a holding chamber for food, more secretion and digestion occurs within the stomach. The stomach can be separated into two regions based on the type of cells and secretion it releases. The oxyntic mucosa is the predominant area of the stomach. Lower in the antrum is the pyloric gland area. All areas of the lumen of the stomach is pitted with deep pockets that form infoldings of gastric mucosa. These infoldings are called gastric pits, at which the base of them lie gastric glands. Gastric pits contain a variety of secretion cells that line their invaginations. Each day, the stomach releases about 2 liters of gastric juices. Gastric juices are predominantly secreted from exocrine cells within the oxyntic mucosa. The 2 liters of gastric juice contains watery mucosa, which is produced from the mucosa cells line the gastric pits and the entrance of the glands. Deeper in the gastric glands are chief cells. Chief cells secrete the enzyme precursor pepsinogen. When activated, pepsinogen will reconfigure to form pepsin. Parietal cells secrete hydrochloric acid and intrinsic factor. Also within the gastric pits are stem cells. These cells rapidly divide and are the parents of all new cells within the gastric mucosa. The cells that are created will either migrate out of the pit to become surface epithelial cells or migrate deeper into the gastric gland where they differentiate to be chief or parietal cells. The stomach mucosa is replaced approximately every 3 days. The main difference between the oxyntic mucosa and the pyloric gland area is the pyloric gland area does not secrete hydrochloric acid. It does secrete mucus and small amounts of pepsinogen. Between the gastric pits, the gastric mucosa is covered by surface epithelial cells that secrete a thick alkalinic mucus that forms a visible layer several millimeters thick over the surface mucosa. This is important in preventing hydrochloric acid from preemptively damaging or killing cells found in the mucosa. Hydrochloric acid, which is released from the parietal cells, aids in 4 main functions within the stomach. First, it drops the stomach acidity to approximately a pH of 2. Second, it activates pepsinogen to pepsin, which allows pepsin to begin its digestive process. Hydrochloric acid, due to its acidity, is able to denature proteins. This means it causes complex 3-dimensional structures of proteins to unfold, increases the amount of surface area which is exposed to the enzymes within the small intestine. Hydrochloric acid also kills many microorganisms within the stomach, which prevents the growth of bacteria in the digestive tract. However, some bacteria does survive and begins to reproduce and colonize within the large intestine. Also secreted by the parietal cells is intrinsic factor. Intrinsic factor is important for the absorption of vitamin B12 within the terminal ileum of the small intestine. Vitamin B12 is essential for the formation of red blood cells. In the absence of vitamin B12, erythrocyte production, it becomes defective and anemia results. Pepsinogen is released by the chief cells. Pepsinogen is an inactive enzyme that, when combined with hydrochloric acid, forms pepsin. Since hydrochloric acid is not formed until it reaches the lumen of the stomach, pepsinogen is also not activated until it comes into contact with hydrochloric acid within the lumen of the stomach. This is important because if pepsin was released in its active form within the gastric glands, it would begin to eat away at the cells that produced it. Pepsin has a self-activating component to it. Pepsin can also activate pepsinogen to its active form of pepsin. This means if pepsinogen comes into contact with either hydrochloric acid or pepsin, it will be transferred into its active form. Pepsin is able to digest proteins within the stomach lumen. Hydrochloric acid comes into contact with these proteins as well and unfolds them, so pepsin has a greater surface area to break these bonds between the proteins. The epithelial cells of the stomach are covered by mucus, which is produced by the mucosal cells. Mucus serves as a lubrication for the stomach to protect it from mechanical stress as food is moved from the fundus to the antrum through peristaltic contractions. In addition to providing lubrication, it also serves as a protective barrier between the contents of the lumen and the epithelial cells lining the lumen. Mucosa is alkalinic, which helps it neutralize the acidity of the hydrochloric acid in the stomach. It also protects the epithelial cells from pepsin, which could break down the plasma membrane of the cell. Other secretory cells within the gastric glands release endocrine and paracrine regulatory factors, which impact the rate of digestion. Vagal nerve stimulation increases the release of acetylcholine. Acetylcholine is the primary controller of the amount of gastric digestive juices released. The release of acetylcholine is going to have a stimulatory effect on the parietal, chief, G cells, and endochromaffin-like cells, ECL cells. An increase in acetylcholine is going to increase the amount of hydrochloric acid secreted by the parietal cells, increase the amount of pepsinogen released by the chief cells, increase the amount of gastrin secreted by the G cells, and it will increase the amount of histamine released from the endochromaffin-like cells. The hormone gastrin, once in the bloodstream, will also stimulate the parietal and the chief cells. In this way, gastrin has an additive effect to the production of hydrochloric acid and pepsinogen. Typically, G cells are stimulated by an increase in protein in the stomach lumen. This increase in protein within the lumen stimulates the G cells to release gastrin. Gastrin is going to be released into the blood and target the parietal and the chief cells within the stomach lumen. This will cause an increase in hydrochloric acid and an increase in pepsinogen released from the chief cells. This will increase the amount of active pepsin within the stomach, allowing for greater breakdown of the proteins. Gastrin also stimulates endochromaffin-like cells to increase the amount of histamine that the cell is releasing. Histamine will act locally on the parietal cells, stimulating it to again release more hydrochloric acid. D cells are stimulated by a decrease in stomach pH, which occurs as food introduced into the stomach is slowly moved into the small intestine. As gastric juices are able to accumulate, D cells are stimulated to increase the amount of somatostatin. D cells, which are found near the pylorus and in the duodenum, secrete the paracrine somatostatin. Somatostatin is going to inhibit the parietal and chief cells, the G cells, enterochromaffin-like cells. This will decrease the amount of hydrochloric acid, pepsinogen, gastrin, and histamine released by each cell respectively. Since histamine and gastrin stimulate the parietal cells, a decrease in gastrin and histamine is also going to result in less stimulation of the parietal cells to further decrease the amount of hydrochloric acid released. When food reaches the stomach, it remains in a semi-solid mass because the peristaltic contractions in the fundus and the body are weak because of the thin layer of muscle mass in this area. This prevents the food from mixing with the gastric secretions. Inside of the bolus, amylase is still in direct contact with the food bolus. This direct contact of amylase and carbohydrates allows carbohydrate digestion to continue within the stomach. Amylase is typically deactivated by acidic environments. However, since the amylase is in the middle of the food bolus and digestive juices cannot penetrate to the middle core of that food bolus, the interior amylase is still able to break down carbohydrates during this time. As peristaltic contractions continue, the food bolus will eventually work its way down into the antrum of the stomach. Here, The food bolus will be fully mixed with the gastric juices, including the hydrochloric acid and pepsin. Protein digestion begins in the stomach due to the pepsin within the antrum. Remember that hydrochloric acid does not actually digest any food. It simply denatures the proteins found within the food bolus to expose a greater surface area to the pepsin molecules. Similar to the mouth, no food or water absorption occurs within the stomach. However, alcohol and aspirin begin to be absorbed within the stomach. The pancreas is located just below the stomach and connects with the duodenum of the small intestine. The pancreas is also connected to the liver from a bile duct. In addition to secreting insulin and glucagon, the pancreas excretes several important digestive enzymes that are fully capable of breaking down all food in the absence of all other secretory enzymes. The acinar cells secrete pancreatic enzymes, and an aqueous alkaline solution is secreted by the duct cells that line the pancreatic ducts. Alkaline solution is rich in sodium bicarbonate, which is used to neutralize the acidic chyme as it enters into the small intestine. Acinar cells secrete 3 categories of digestive enzymes: proteolytic enzymes, pancreatic amylase, and pancreatic lipase. Proteolytic enzymes digest proteins. The first proteolytic enzyme is trypsinogen. When released from the acinar cells, trypsinogen is in an inactive form. This prevents trypsinogen from damaging the cells around it. Once it enters into the duodenum, it is combined with enterokinase, which activates the trypsinogen into trypsin. Like pepsin, trypsin is a self-activating enzyme. This means when it comes into contact with trypsinogen, can activate the trypsinogen to the trypsin. The other proteolytic enzymes secreted by the acinar cells, procarboxypeptidase and chymotrypsinogen. When procarboxypeptidase and chymotrypsinogen come in contact with trypsin in the duodenum, they are activated into carboxypeptidase and chymotrypsin. Again, neither of these enzymes are released in their active form. They must be activated by coming into contact with trypsin within the duodenum. Since the saliva amylase becomes mostly inactive once the food reaches the antrum of the stomach, the secretion of pancreatic amylase continues the digestion of carbohydrates, continuing to break down polysaccharides into disaccharides. Because the pancreas secretes bicarbonate, it neutralizes the acidic chyme within the small intestine and allows amylase to remain in its active form. Pancreatic amylase is released from the acinar cells in its active form because these cells do not contain polysaccharides. Pancreatic lipase is incredibly important because it is the only enzyme released in the digestive system that is able to break down triglycerides to monoglycerides and fatty acids. Pancreatic lipase, again, is released in its active form from the acinar cells because there is no risk of pancreatic self-digestion by lipase. The secretion of pancreatic aqueous alkaline solution from the duct cells within the pancreas are incredibly important to the functions of the enzymes released by the pancreas. Enzymes function best in a slightly alkaline or neutral environment. Yet the chyme that enters into the duodenum is highly acidic. The alkaline solution quickly neutralizes the acidic chyme and allows enzyme activity to continue unhindered. The aqueous solution is by far the largest component of the pancreatic secretion. One way in which pancreatic secretion release is monitored is by the release of secretin. When the duodenum becomes too acidic due to too much gastric emptying, it increases stimulation to the S cells, which are also found in the duodenum. The S cells increases its release of secretin. Secretin travels through the bloodstream to its target organ, the pancreas. Once in the pancreas, secretin stimulates duct cells to increase the amount of alkaline solution it is producing and releasing. This will result in more alkaline solution being released into the duodenum to neutralize the acid in the duodenum. Secretin will also impact the stomach by decreasing gastric emptying, by decreasing the strength of contraction within the stomach. By decreasing contraction strength, less chyme is able to move through the pyloric sphincter into the duodenum. This decrease in gastric emptying also decreases the amount of new acidity introduced to the duodenum. Another important regulator of pancreatic secretion is cholecystokinin. Cholecystokinin, or CCK, is stimulated for release when there is an increase in fat in the duodenum. This increases the stimulation of the mucosa of the duodenum, which increases the release of cholecystokinin into the blood. CCK travels to its target organ, the pancreas. Once in the pancreas, it targets the acinar cell to increase its production and release of pancreatic enzymes. Similar to secretin, increased fat in the duodenum results in a decrease of gastric emptying by a decrease of the strength of contraction. By the time chyme reaches the large intestine, digestion has been completed. This means that the large intestine does not secrete any digestive enzymes. However, the large intestine does secrete an alkaline mucosa. This alkaline mucosa serves four main functions. First, it protects the large intestine from mechanical and chemical injury. It lubricates to facilitate the movement of feces throughout the large intestine, and it neutralizes acids produced by local bacteria. Because movement within the large intestine is slow, bacteria have time to grow and accumulate in the large intestine. In addition, the large intestine does not secrete antibacterial agents. This allows surviving bacteria to continue to grow and thrive in the large intestine. This bacteria is not only typically harmless, but actually is incredibly beneficial. It enhances intestinal immunity, it promotes motility, helps maintain mucosal integrity, and makes nutritional contributions such as synthesizing absorbable vitamin K and raising acidity, which promotes the absorption of calcium, magnesium, and zinc. Each day, approximately 500 milliliters enters into the large intestine. However, only 150 milliliters per day are excreted in fecal matter. In the large intestine, water is reabsorbed, sodium is actively transported out of the large intestine, and chloride ions passively follow down its electrochemical gradient. A small amount of vitamin K, potassium, calcium, and magnesium are also absorbed from the large intestine.